Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. I'm your host, Eddie Avil, and today I'm delighted and honored to have with me Mr. Petr Šulc, who leads the Šulc Lab, an interdisciplinary research group, and is an assistant professor at School of Molecular Sciences and the Biodesign Institute, Arizona State University, which is affiliated with the Center for Biological Physics, Center for Molecular Design and Biomimetics, and Center for Biocomputing, Security and Group and Society. The lab employs methods of statistical mechanics and molecular simulation to study DNA and RNA, with a particular focus on DNA and RNA nanotechnology. So, Peter, really appreciate you taking time and being part of the podcast. Would be great if you could start with, priming the audience and maybe explaining what nanotechnology is.
Okay, so nanotechnology refers to size of nanoscale. So nanoscale means 10 to the minus 9 meters. So that is the basically length scale at which molecules interact with each other. And the field of nanotechnology is based on designing devices and structures where you control their operation at nanoscale level. So that would be basically the sizes at which living organisms internally function. So you think about structures inside your cells, how your cell operates. If you would compare a living cell to a factory, nanoscale would be the typical size of the machinery at which your cell operates. And we in the field of nanotechnology basically try to design structures and devices that would allow us to interact at this scale for building things which can have therapeutic applications, but also to do completely synthetic designs where, again, you have control over individual nanoscales.
So nanotechnology was something which was kind of push-driven by Richard Feynman's lecture, lots of room at the bottom. Yeah, and then Eric Drexler, he wrote a book called Engines of Creation, where he talks about a nano assembler. So you spoke about nanotechnology, and you said that with when you're kind of tinkering around with these nanoscale molecular atoms, the goal and the vision is to kind of create things out of it. So explain that concept and how do you go about building a nano assembler? And when you have a nano assembler, what is it that you would be able to do with it?
Yeah, so that is correct. The field indeed is considered to be started by this visionary talk by Richard Feynman in 1959, I believe. But in reality, that talk went largely unnoticed until it was sort of rediscovered by Eric Drexler, who really popularized the notion of the field of nanotechnology, especially with the book that you mentioned, Engines of Creation. So the book, which was published in, I believe, in 1988 or something like that, was quite visionary. And he envisioned the future which would be based on nanomaterials and nanoassemblers. And in particular, he really focused on this idea of nanobuilder. So in the case of, again, imitating biological systems, he envisioned we would basically build a machine that would be able to build structures directly from individual atoms, putting them together. Which, if you were able to have such a machine, you would basically, what you probably know from Star Trek movies and TV series, they have this device called I believe replicator, or what is it called, where you basically just say that you want some object like a glass of water and it creates it from you from empty space by presumably assembling individual atoms to create whatever you want. And Eric basically envisioned that we would build these assemblers in multi-scale level, that you first build one class of larger assemblers, which then assemble smaller assemblers, which will then eventually get you to the atomistic level. And they would, among other things they can assemble, they can actually assemble also their own copy. So in his vision, you would get these assemblers making other assemblers, and they would eventually trigger exponential growth of them, which would allow you to build any material and any structure you want. To be fair, this vision hasn't been met with only approval in the scientific community at the time, and famously Eric Drexler had a long-standing, I would say, discussion or argument with Nobel Prize laureate Richard Smalley, who basically thought that it's impossible to make a machine where you would really manipulate individual atoms and put them into the place where they are supposed to be. This argument is referred to as, sticky fingers, basically boiling down the idea to the fact that once you pick up a single atom, it attaches to the whatever structure it's interacting with the atom, and you can't really selectively detach from the atom and deposit it to just one possible position easily. So I would say that the field is still not entirely sold on the idea of building a nanoassembler on atomistic level, as Eric Drexler originally envisioned. But it's, of course, very much fascinated with the potential of building a self-replicating mechanism, or even building the things that Eric envisioned at atomistic level, even achieving something where you can really manipulate individual molecules like DNA molecules or parts of proteins and put them into one place. That is still very much a focus of many labs in the field.
Where do we stand with the cutting edge of innovation? And because everything is made out of atoms, the entire world is made of atoms. So in the future, if a nanoassembler is possible, a self-replicating nanoassembler, then in theory, we would be able to possibly maybe print out, build out anything or everything that we can imagine, right? Could you elaborate on this? What would be if and when we get onto building a machine like this?
So that is indeed the premise with a very huge if. And there are certainly many prominent scientists who do not think that such machine is physically possible. So, but if it was assembled, you would really be able to scale up production of almost anything, right? Then, of course, the question comes, there's no free lunch in nature, right? We are always limited by second law of thermodynamics and by the first law, which is the conservation of energy. So it's still not clear how energetically costly the operation of such machine would be. So I'm still quite hesitant about selling to this idea that we would have an nanoassembler that will just make its copy and assemble anything you want, material of any property or any structure. That I think might not be plausible. But even if we are not really able to manipulate and self-replicate nanostructures at atomistic level, which is basically scale 10 to the minus 10, that's even one order of magnitude below nanoscale. If we are able to arbitrarily manipulate molecules at nanoscale, that is 10 to the minus 9, already that machine at that level or devices have great potential in basically interacting in any way you want with biological systems, and also constructing new materials with properties which we cannot construct otherwise. So even the ability to construct a material where you control the distances between individual components, which can be larger group of atoms, it doesn't have to be a single atom, but even larger group of atoms or molecules, where you can position them at distances which you control with nanometer precision, still has a huge potential in terms of novel material properties.
Can you explain your research focus on DNA and RNA biophysics and nanotechnology?
In bio-nanotechnology, we use existing molecules from nature. So we basically take DNA, RNA, or proteins, which are sometimes directly taken from natural components. Sometimes we synthesize them from individual building blocks, which in the case of DNA and RNA, those are individual nucleotides. In case of proteins, those would be basically you can translate protein of any choice you want from provided template. And if you are smart about how you design your sequences, you can then fold them or have them self-assemble into a given shape or structure. So the concept of self-assembly is based on individual units randomly moving around until they bump into each other and form a bond. So if you think of building stuff with Lego blocks, which is my favorite analogy, what you are, of course, used to is taking each individual Lego piece and putting it according to the manual into one particular point. But this is not how structure makes things, right? Structures in nature, including ourselves, are built by self-assembly. So there will be individual molecules, each traveling through chaotic motion into a particular point when they are needed. So the analogy would be that you take your Lego blocks, you put them all into the washing machine, and just by randomly spinning them, they would all fall into the pieces as they need to be, and you would get the structure out after a few minutes out of your washing machine. It of course doesn't work with Lego blocks, because Lego blocks are not designed to self-assemble. They are designed to be top-down assembled. You need an external input like your hand, which puts them in place where they should be. And at Bio-Nanotechnology, we basically try to design systems which are capable of self-assembling into the structure and shape that you want. So for a long time, and we are to some extent still in that era, a lot of the research in this field was just about proof of principle. It wasn't necessarily looking for a particular application, but just to understand better the rules, how to design a DNA molecule or RNA molecule so that they would assemble into a particular structure. Now, for listeners who are not too familiar with DNA, RNA, and proteins, so DNA is a basic molecule of life. Every single living organism on Earth stores their genetic information in a DNA double helix, and that code is read by the machinery inside cells, produces RNA, proteins, and basically organizes function of every single cell. Now, you can also synthesize small bits of DNA chemically. So you can design the sequence, and then you mix multiple of these synthesized DNA molecules together, and they will form different connected double helical regions. So instead of having one huge, super long double helical structure that you have in your genome, you'll have a smaller compact structure which consists of multiple connected double-stranded domains together. That is the basic idea behind DNA nanotechnology. The younger cousin of DNA nanotechnology uses RNA, basically performing the same function. And in recent years, especially thanks to generative AI, that would be basically just in the last year and a half, we have seen a great progress also in people being able to design proteins that would fold into a desired shape.
How awesome is this, Professor? Because what you are trying to say over here is obviously, I mean, nature self-assembles, but there's something magical going on, is what you're claiming, is that these molecules or even atoms, if you kind of throw it, the analogy is that you gave is, you mix it in a washing machine, and they have these innate capability of forming into these complex structures. So it's not the humans you're saying is actually compiling these atoms, atom by atoms, into a specific structure, but you're saying here that nature somehow has this ability of, even if you throw these random atoms or molecules in whatever petri dish or a device, these molecules and atoms have a capability of forming these natural structures of sort. So in theory, I mean, if you kind of understand what the self-assembly is doing, then we possibly will be able to build throw out these molecules or atoms, and it'll be able to kind of come out with these extremely interesting molecular structures. Could you delve into that a little bit more? When you say that, when it self-assembles into certain shape and structure, what could be the possible applications of this, when
you want to understand
shapes and structures?
What the field is trying to do currently is to come up with more complex devices and more complex structures. So how do we quantify complexity? So one thing is just the overall size. So for example, we want to do structures which are micrometers by micrometers in size, but they are built from nanoscale sized blocks. So that would mean that we can position in three-dimensional space different functional molecules with nanoscale precision, but the overall size of the thing would be in several microns. So yet again, if I make the analogy with biology, a human is maybe 1.5 meters tall on average, let's say, but it all comes down from individual processes that happen at nanoscale level in each individual cell. So inside the cells, you have these machineries which organize not only the insides of the cells, but also how the cells are positioned next to each other. And that is a whole coordinated system at the end. So in the very, very simplified, dampened down, striving in one of the field of nanomanufacturing is just to be able to build larger structures from individual nanoscale blocks. And yet again, as I say, a lot of people in my field just focus on the proof of concept. Like the structure that they try to build doesn't need to have any application. It can just be a simple cube where you can attach a fluorescent molecule at certain distances and just show under the microscope that you can make like letter A out of them. Just that, no particular application.
Right.
Another direction is computing. So can you actually make logic operation at nanoscale? So again, bacteria, in a sense, computes, right? It swims, it gets sugar, then it senses the direction in which there's more sugar, so it tries to swim in the direction where it can get more nutrients. So it does some internal computation as a function of its surroundings. And there are different strategies that people try to do at nanoscale to also realize computation. So for example, circuits that sense whether a certain molecule is present or not in the solution. If it is present, they give off a fluorescent signal. If it is not present, they don't give off a signal. So again, a lot of work has gone just to understanding how to design these circuits so they are robust and they can operate in chaotic environment. But then there are possible applications, like what we are trying to build that would be useful. So one direction is, of course, therapeutics and diagnostics. If we are actually able to realize these computations inside cells, then you can envision a future where you have like some intelligent, well, I probably shouldn't use the word intelligent, but nanorobotic device, which is capable of getting inside cell and evaluating its environment. So it can decide, oh, in this particular cell, there's a mutation in DNA, which might cause cancer. So let's kill this cell. Another thing is diagnostics. So you can have devices which can sense whether in your blood serum you have virus or not, for example. Another direction is leveraging the effect that you are able to position molecules at very particular distance. So there are already some companies that have been started by colleagues in my field, which leverage this mechanism for interacting with immune system. It turns out that different cells of our immune system, they have plenty of receptors on their surface. And many of the therapies are based on engaging with these receptors. But all the drugs which are currently on market would typically only target one receptor at a time. If you have a nanostructure where you have multiple of the molecules which can bind to these receptors positioned at certain distances, it turns out that you can engage multiple receptors on the surface of the cell, and that can increase the activity, or for example, lead to different type of activation that would be able to achieve with just a single receptor. So there's a huge potential for all these techniques in medicine, therapeutics, diagnostics. Other direction that people are taking is building completely new materials. So I already mentioned it's super appealing, not only at atomistic level, but also positioning nano-sized material blocks in 3D space. So just the fact if you are able to take two different materials and arbitrarily position them into a block where one region has titanium in it, other region has just silica in it, those materials would unblock completely new ways how we can make materials that interact with light, for example. You could make new detectors. You could even possibly realize logic gate operations directly on light, which would be much more energetically favorable. could basically unlock the field of what is called optical computing, where instead of using transistors to logic gate operation, you just have these specially designed materials produced maybe by self-assembly, maybe by these molecular printers that Eric Drexel envisioned at atomistic level, but they would be maybe at nanoscale level. And it would really unlock on individuals engineering.
Here we are, tinkering around with nature and the cusp of kind of trying to figure out. You clearly said that, building a living organism, we are very, very far away. But where we are at this point of time is building complex structures. How do you go about doing this? Because you spoke about self-assembly. So in theory, I mean, if you're able to kind of, put these atoms together, because it self-assembles, because that's the way the universe, the mechanism of things are. How do you get from a simple structure to a complex structure to a living organism? What would be the process?
And the answer basically is that it's super difficult. Biological molecules consist of many different atoms arranged in very complex molecular shapes. They have very complex interactions with each other. So it turns out that the more accurately you approximate them, the more difficult the computation gets. So if you go down to exactly quantum mechanistic level, which is like the most detailed, most accurate interaction description between molecules and atoms, you can maybe only accurately describe system which consists of tens of atoms, right? The typical nanostructure size that we can produce in lab trivially today has around 13,000 nucleotides in it, and each nucleotide is between 20 to 30 atoms. So there's several orders of magnitude above what we can accurately calculate with just quantum mechanical approach. So what you do, you simplify further. You forget about quantum mechanics and you basically describe interactions between atoms with Maxwellian electrostatic equations. Turns out even those are quite difficult to solve. And the best models out there can maybe simulate processes which take maybe a few microseconds. But if you look at biological systems, typical processes take milliseconds and longer, right? It can take several hours for one cell to divide. It can take minutes for proteins to be produced and correctly folded, etc. So basically, we have this timescale problem that the more precise we go, the shorter times we can simulate. And it's not that the computers will be powerful enough anytime soon, right? So with the Moore's law, which it's still a question how much longer it can hold, we double the speed every 18 months or so. But to go from 1 microsecond to hours to days, that's still years and years away. And these models still have their imprecisions. So what my group is, for example, trying to do is to come up with a multi-scale approach. We basically try to simplify the simulations of the systems that we study as much as possible so that we can simulate larger systems and long timescales. And we always try to make sure that the details that are relevant to the particle systems that we study are still accurately captured. But we do not worry about the other details which are maybe not so important for the particle behavior that we want to understand. So for example, I mentioned some of these nanostructures that can perform computation inside cells. Right? So in that case, we just want to understand how these reactions depend on different parameters of the system. Like if I design the DNA strand in this particular way, what is the probability it's going to malfunction? But what is the probability that my AND gate is going to give me output 1, even if the output is 0 and 0 in terms of the molecular representation? So if I'm modeling such process, I don't really care how does protein interact with DNA. So I just assume let's ignore all the other proteins that can be in the cell for now, and let's just focus only on the interactions between individual DNA strands. So this process is called coarse-graining, and with these approaches, we've been able to scale up the simulations up to systems consisting of several millions of nucleotides. And we've been actually able to study some interactions which would take minutes to hours in the lab. and guide experimental designs based on that. But of course, those are still systems which, as I said, we ignore many biologically important factors. So that's okay when you work in the lab where you just have water into which you add the DNA molecules and nothing else is there. But once you try to take these nanostructures that we designed this way and put them inside cells, sometimes they completely malfunction and we have no idea why, because we, of course, left out many important interaction details which happen in cell. And then again, you need a synergy of better molecular experiments. So the better equipment we have, the more carefully we can study individual molecules movement inside cells, then we can get better understanding what are other things the nanostructure can be interacting with. That gives us some understanding into how to develop better models. And then basically iteratively we can improve our design pipeline, have better models that can simulate more complex environments, and then design structures which are capable of more complex behavior. But it's a process which is taking, of course, very long time. Some people are hoping that the recent advances in generative AI will really speed up the development of these new models, and that's one of the most exciting venues that this field might be taking. But we are still at the beginning, so we are not really sure how far we can go, how much better models we can build with AI that we were not able to build just by our rational intuition so far.
You spoke about, the nanotech applications, right from with those self-assembly, your simple shape and structures to, simple structures to complex structures, to nanoscale constructions, possibly drug delivery systems, possibly maybe nanobots, nanophotonics, biocomputing, and so forth. What's the timelines for this? When do you think that we would be able to see these applications in the market? Besides yourself, who are the other peers who you think are really driving this conversation forward?
So in terms of the protein designs, the definitely towering figure of this field is David Baker. Just today, actually, a few hours ago, I saw an announcement that he sold one of his companies for $1.1 billion. And that one was used to design a protein-based vaccine against COVID. So in his case, he basically has a huge group which builds both software and experimental realization of protein nanostructures. And he basically built a protein cage, which is used as a vaccine. So the proteins basically are injected into the bloodstream of the patient. The proteins on this cage display the particle adjuvant, which is derived from SARS-CoV-2, and your body develops immunity against it. So this is one example of basically using it as a cancer vaccine already. And that is already in market. That vaccine has been approved in South Korea, I believe. And I can see many other developments for vaccination against other diseases, maybe against cancer, personalized vaccines in this direction. So another exciting area which I have seen, and there's a company called Switch Therapeutics, for example, is to try to bring this logic gate computation into therapeutic approach. So currently, almost all, if not all, FDA-approved drugs would be typically small molecules which go into the bloodstream and they bind to the particular protein which is known to be associated with disease. So there's no logic gate operation there. If you see the protein, it binds to it, and that's it. And some of these companies, we try to leverage some of the techniques of logic gate computation at molecular scale, try to design devices which get into the cell and say, okay, if only this particle molecule is present, for example, some RNA which is associated with a disease, only then I will try to, for example, knock out this gene which causes the disease. So it will be specifically targeted to only affect one particular cell type and do only one particular action if the condition is satisfied. And this is based on ideas from DNA and RNA technology, how to compute using DNA interaction strands, which is a method called strand displacement. It's a simple mechanism which can scale up to quite complex behavior. Another directions that I've seen in DNA nanotech is, for example, the work of a group of Hendrik Dietz at University in Munich, which designs DNA nanostructures which are able to capture virus. So he basically designed, has a company that started 1 or 2 years ago, I believe. So they design nanostructure out of DNA, which on its surface has antibodies which bind to the surface of the virus. We all know that virus mutations are a big problem, right? Like the original virus SARS-CoV-2 has quickly mutated, and after a few months, the antibody drugs that were developed for the original alpha variant were no longer effective against the virus because its surface protein mutated. So the binding of the drug was clearly decreased, and this was no longer effective against the virus. But if you put multiple of these weakly binding drugs on top of the DNA nanostructure, each of them individually binds very weakly. But since you have the entire structure covered with them, they will still stick very strongly to the virus. Think of it that you basically try to glue something on the wall with just one little piece of glue, it will not hold the picture. But if you have many, many different tiny dots of the glue and you slap it on the wall, it will still hold, even though each individual on its own wouldn't be strong enough. So you can design nanostructures like this, which can completely envelope the virus, and it prevents it from being able to infect cell completely. So there is a huge consortium now in Munich trying to further develop these technologies, for example. And the last thing I think is on the horizon, pretty reasonable future, like maybe several years' time, at least into the first applications, would be converting biological interactions into electric signals. So biological interaction, for example, means two proteins interacting. Or you have your protein, which is designed or derived from an antibody, which you know binds very strongly to virus protein. But the event of the protein binding to the virus happens at very quick time scale, and it's not visualizable easily, right? So if you have this protein in your blood, you want to test even very small concentration of it. And you would like to translate the effect of the protein binding to your probe into an electric signal. And this is another very promising avenue which the field is taking. So basically being able to detect very small concentrations of different biological molecules from biological sample, for example, drop of blood or cell taken from the patient, and convert it into a signal whether that particular molecule we are after is present in the sample or not. It can be for diagnosing viral infection, diagnosing tumors, etc. And there are already companies that are trying to leverage some of the technologies I talked about, like DNA or proteins, in this direction.
Really, really appreciate you taking time and being part of the podcast. You spoke about, role of generative AI in AI, in RNA, and that you saying that's going to like really, really help. I mean, what's your future vision? The approach that you've taken, your multi-scale approach. How do you think that's going to pan out in the future? What's the moonshot, and what comes next for the cell clip?
I think synthetic cell is one of the big moonshots. Having basically an enclosed environment which would be, to some extent, carry out the functions which you only associate with cells. So something which could maybe move in certain direction, like a bacterial swimmer, but it would be completely designed. So chemically powered locomotion, a system which can take chemicals from its surroundings and process them into some new product. So like nanoscale chemical factory, if you will. Those are like some of the big challenges that people are trying to come up with. Systems like going even more further in terms of the complexity, like systems which are capable of some evolution based on their surroundings. So something which can make mutation to its own architecture and change as a result, right? So if you can build a machine which can automatically modify its parameters as a response to its surroundings, that would be a major, huge deal, right? Because then you can really evolve machines for a particular function. But there you need to have some sort of, if not self-replication, at least some feedback where you have an assembler which evaluates the structure and assembles a new one based on the performance of the previous one. So these are some of the really great challenges. And in terms of where generative AI and modeling can help us is that the better we have simulations of these systems, the easier it gets, right? If you do experiment, it takes hours or days to mix all the chemicals, image them, it gets very messy. The more accurate simulations of the systems you have, the longer timescales you can simulate, the more of the design you can offload to just simulation, right? And again, going back to my original analogy with planes, when Lockheed Martin designs the F-35, it's not like they build 1 million different wings and try to fly a plane with all the different wings they designed to see which one is the best, right? They have very good algorithms that simulate the wind flow around it. Then the best design they take to the wind tunnel, and only then they would actually build a jet that flies, right? And I think we are still in our technology missing to having like really good computer world model that would allow us to design very quickly all these complex things. But once we have a very good computer representation of the real world, then you can imagine you have like AI, like the AlphaGo, which was playing against itself, the game Go, until it became unbeatable champion. You can then have generative AI, which just keep generating nanostructures and simulating their behavior until you get something which really performs the task like self-replication really well.
Right. I hope that we get into the world, because the pace at which technology is accelerating and it's all converging, I'm sure hopefully in the next few decades, maybe all those things will come together where it'll give you the tools and the capabilities where the self-assembling really goes from simple to complex structure, and maybe someday, maybe like a living organism possibly. So wish you and your team the very best. And to my listeners, if you like what you see any other, please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you. Thank you, Peter. Really appreciate it.
Thanks very much. It was a pleasure talking to you.